How do you feel after your surgery, Miss Wong?
I feel a little bit woozy.
That's probably because you lost a lot of blood, but I have your transfusion right here.
Is that milk?
Oh, yeah, that's definitely human milk. I think it's my mom's.
Why don't you just use blood?
I think I have some blood right here.
Is it human blood?
Ew, no, it's sheep blood. Some of the first successful transfusions were animal-to-animal experiments by William Harvey. Later, physicians attempted to transfuse animal blood into humans. In 1667, Jean-Baptiste Denis transfused sheep blood into a 15-year-old boy who survived. James Blundell was one of the first physicians to practice human blood transfusions. After experimenting with dogs, Blundell successfully transfused blood from a husband to his wife in 1818. In the 1870s, scientists began searching for an alternative for blood. Animal and human milk was briefly used as a blood substitute, but adverse reaction led to the increase in saline as a blood alternative. In 1901, the three blood groups, A, B, and O, were discovered by Karl Landsteiner, the father of transfusion medicine. He also found that blood transfusions were only successful when the blood types are the same, or else the blood agglutinates and gets clumpy. In 1927, Landsteiner and Philip Levine discovered three more blood groups, M, N, and P, which were then used in paternity cases to determine who the father of a child is. Before 1940, not all transfusions were successful, even when the blood types matched. Karl Landsteiner and Alexander S. Weiner discovered the Rh factor, which was another antigen on the surface of red blood cells. If the blood cell has the antigen, it is considered Rh positive, and without the antigen, it is Rh negative. It was named Rh factor because the blood was from rhesus monkeys. Blood transfusions help save those who have lost a lot of blood, and it especially caters to hemophiliacs.
Hello, my name is Dr. Humphrey Davy. My friends call me Humpy. Are you ready for your dental procedure today?
Yeah.
So we are going to need to anesthetize you for today's procedure. I will be using nitrous oxide.
Isn't that deadly?
We'll find out. Nitrous oxide is also known as laughing gas. I haven't turned on the gas yet. In 1772, Joseph Priestley discovered nitrous oxide gas, which he called dephlogisticated nitrous air. Humphrey Davy found that inhalation of nitrous oxide could relieve a conscious person from pain in 1799, but the nitrous oxide, or laughing gas, was not used as an anesthetic for another 44 years. It was, however, used as a recreational drug, and Davy himself became addicted. In 1846, William T.G. Morton, a dentist, publicly demonstrated at the Ether Dome in Boston that inhaled ether can be used as an anesthetic. He claimed to be the discoverer of anesthesia, but Crawford Long has records of using inhaled ether as a surgical anesthetic as far back as 1842. An alternate anesthetic, chloroform, was discovered in 1831 by Samuel Guthrie. James Simpson experimented with Guthrie's chloroform in 1847 while looking for a replacement for ether. Simpson began administering the chloroform to patients during childbirth, and chloroform began to replace ether as an anesthetic. The downside to chloroform was the tendency for patients to die after inhaling it, but the number of deaths were greatly reduced when John Snow created an inhaler to ensure the proper dosage of chloroform was administered. Another substance that has been used as an anesthetic is cocaine. In 1884, Carl Collard demonstrated the tissue-numbing quality of cocaine in eye surgery and was nicknamed Coca-Collar.
Good day, Sarah Neems. I require some milk from your dairy cow.
Oh yes, of course. Be careful though. I got cowpox from my cow Blossom here. The good news is I can't get smallpox now. Well, did you want your milk?
Oh, yes. Thank you. If I'm going to test this hypothesis, I will need a test subject. You there!
Yes, sir?
What is your name?
My name's James Phipps, sir. I'm 8 years old.
Would you like to make a lot of money?
What?
Alright. If I could just see your arm. Now I'm going to take this sample from you and transfer the cowpox to you. This will only hurt a lot.
Why? That crazy doctor poisoned me!
Doctor!
Yes?
I got smallpox.
Congratulations. You are a perfect test subject for me. Now, what I'm going to do is take her smallpox and infect you, knowing that you will be safe because of your recent bump with cowpox.
What?
I just have a hunch. Don't worry about it. Give me your arm.
What? No! I'm gonna die.
Goodbye. See you in a week.
Variolation, or deliberately infecting a person with smallpox in order to prevent more occurrences of the disease, was widely used in the early to mid-1700s. Although people were protected from further smallpox infection, there was a 2 to 3% fatality rate associated with variolation. In addition to this, variolated individuals could pass the disease on to others. When Edward Jenner heard the rumor that cowpox would also protect a person from smallpox, he decided to conduct an experiment. Jenner infected an 8-year-old boy, James Phipps, with cowpox from the hand of a milkmaid named Sarah Neems. The boy made a full recovery, and 2 months later, Jenner inoculated James Phipps with matter from a fresh human smallpox sore, and the boy did not contract smallpox. In 1800, Benjamin Waterhouse brought the vaccination to the United States, and he performed the first vaccinations in the U.S. In an effort to increase vaccine production, lymph from calves infected with cowpox was harvested and used as a vaccine in 1877. In 1980, smallpox was declared eradicated. Polio was a big epidemic at the beginning of the 1950s. Thousands of children were dying or seriously paralyzed. After receiving a grant from the National Foundation for Infantile Paralysis, Jonas Salk began working to discover the vaccine for polio. Salk developed a killed virus vaccine and began running human trials in 1952. By 1955, Salk's vaccine was declared safe and effective, and after 2 years, 100 million doses of the vaccine had been distributed throughout America. Because of vaccines, the eradication of smallpox and elimination of other diseases has occurred. Also, death rates have dropped greatly. Vaccines have been integrated into society through the mandatory vaccines that school children must get, making it a commonplace procedure in American life. Life expectancy is also extended because of vaccines. In addition, the use of vaccines has helped relieve pressure off of negative outcomes of other medical practices, such as the development of antibiotic-resistant bacteria due to the overuse of antibiotics, showing the interaction and relationship between various medical practices.
Wow, autopsies sure are fun, but they're also pretty messy. Oh, I think my 4 o'clock's here. Come sit down. You ready for abdominal surgery? Alright, here we go.
Skrobenje, to się teraz äta.
Who are you?
Josef Lister.
And why are you interrupting my abdominal surgery?
Vi skott ifrån. I skibo eller våta.
What?
I refito eller våta.
Why?
Griposkebo!
Wait, Dr. Lister, aren't you British?
Johnny ho!
The germ theory of disease states that some diseases are caused by microorganisms or pathogens that are too small to see with the naked eye. Pasteur conducted a series of experiments throughout the 1860s to prove that bacterial growth came from biogenesis, which states that living things come only from living things, and thus disproved the outdated theory of spontaneous generation, which declared that inanimate objects could give rise to living organisms. Cook's postulates, developed by Robert Cook, outlined 4 different guidelines to identify pathogens. His postulates were, number 1, the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. Number 2, the microorganism must be isolated from a diseased organism and grown in pure culture. Number 3, the cultured microorganism should cause disease when introduced into a healthy organism. Number 4, the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. However, the first postulate was abandoned when Cook discovered asymptomatic carriers of cholera. The person could carry and transmit the disease but still remain healthy. In the 1870s, Joseph Lister discovered that carbolic acid solution swabbed on wounds would reduce the frequency of an infection. He urged surgeons to wear clean gloves and clean their surgical tools to prevent the spread of the disease. Because of his introduction of sterile surgeries, Lister is known as the father of antisepsis.
DNA was first discovered in 1868 by Friedrich Miescher when he noticed a microscopic substance, which he called nuclein, in the pus of discarded surgical bandages. Albrecht Kossel isolated the 5 primary nucleobases of nucleic acid. The base, sugar, and phosphate nucleotide unit was identified by Phoebus Levine in 1919. William Ashbury's X-ray diffraction patterns in 1937 showed the regular structure of DNA. Erwin Chargaff noticed the connection that the amount of adenine to thymine is usually equal, as is guanine to cytosine. This was known as Chargaff's rule. In 1952, Rosalind Franklin and Raymond Gosling published their X-ray diffraction data that was later used by Watson and Crick to determine the structure of DNA. Watson and Crick suggested the double helix model based off of Photo 51 from Franklin and Gosling. The significance in the discovery of DNA is more than just an accumulation of information. The real importance was the potential for growth in all aspects of biological science. For example, the knowledge that DNA is inherited by offspring could aid in the study of heritable genetic diseases. There was no one true discoverer for DNA. Each scientist built off of the work from others until they learned as much as they could about DNA. This shows how progression in medicine occurred. Also, the research in DNA was carried out by scientists from various parts of the world, displaying how medical and scientific information was shared, allowing for growth.
Alright, Noelle, you know why I called you into my office, right?
Did you get my X-ray results back?
So, are you aware that there actually was a knife lodged into your forehead at the time of the X-rays?
I'm really bad at cooking.
So did you jam a fork into your foot before you came in that day?
I like forks!
So are you aware that there actually is a miniature Eiffel Tower lodged in your hand?
I had an accident at the miniature golf course last night.
Why would you put a gun there?
That one you know why.
In 1895, Wilhelm Röntgen, a German physicist, discovered the X-ray. While working with a cathode ray, he found that under certain conditions, it could cast shadows of solid objects on pieces of film. Röntgen recognized that an X-ray could be used to identify bone structures. The very first X-ray done on a human was an image of his wife's hand. The X-ray also came along with many downsides, including the risk of cancer, burns, and hair loss. With this new device, broken bones could be identified without using invasive methods. It also allowed for new inventions like the shoe store fluoroscope, which let customers view an X-ray of their own feet. They were widely popular from the 1920s until they were prohibited in the 1970s because of the high levels of radiation that they emitted. X-rays symbolize not only a medical advancement that helped diagnose many people, but also a social transformation towards novel products centered on fun rather than practicality.
Hi, I'm here for my full body MRI.
Alright, let's get you started. So we'll be putting you in the MRI machine and I'll be viewing the images from a separate room. Alright, Noelle, just keep still and I'll be monitoring the images through my computer here.
Do you guys see anything?
Oh my god, what is that?
It's my baby. As of today, there's an estimated 25,000 MRI machines in use worldwide. MRIs use medical imaging techniques used in radiology to investigate the anatomy and function of the body. An MRI works by forming a strong magnetic field around the area that is going to be imaged. The rate at which excited atoms return back to equilibrium state creates the contrast between different tissues. The main function of the MRI is to diagnose and stage diseases. The history of the MRI dates back to 1952, when Herman Carr created the first one-dimensional MRI image. In 1971, Raymond Damadian discovered that tumors can be recognized using nuclear magnetic imaging. Damadian thought that the machine could be used to diagnose cancer, but the results proved to be too unreliable to use as a diagnostic device. The first ever MRI machine was created by Damadian in 1973, and his patent was issued in 1974. Carr's idea was expanded by Paul Lauterbur, who developed a way to generate 2D and 3D MRI images. The use of the MRI has had tremendous impacts on society. It has allowed for doctors to be able to diagnose fatal diseases early. This means that many more lives have been saved due to the MRI.
Alexander Fleming discovered penicillin in 1928. While working in his lab, he put Staphylococcus into a petri dish and left it open overnight. When he returned the next day, a blue-green mold had appeared in the petri dish. He then noticed that the mold had killed off the bacteria surrounding it. He named the mold penicillin. Penicillin proved to be ineffective against gram-negative bacteria and fungi, and Fleming did not believe that the antibiotic would last long enough in the human body to kill bacteria, so he stopped studying it after 1931. Howard Walter Florey is credited for being the first to use penicillin as a medicine in 1939. Penicillin has opened a great door of opportunity for doctors. It has allowed for the treatment of several diseases as it is a broad-spectrum antibiotic. Penicillin is also the basis of production for today's antibiotics, showing how much of an effect it has on the scientific community. HIV, or human immunodeficiency virus, is a disease of the immune system that attacks the host's T cells. The late stage of HIV is referred to as AIDS, or acquired immunodeficiency syndrome. A person is said to have AIDS when the number of CD4 cells drops below a certain point. Although there is no cure or vaccine, antiretroviral treatment can slow the disease. The United States Center for Disease Control first recognized AIDS in 1981, and by 1989 there were 100,000 cases in the U.S. The initial AIDS cases were mainly injecting drug users and homosexual men. The 4-H disease was coined by the CDC because mostly Haitians, homosexuals, hemophiliacs, and heroin users were affected. The general press even began using the term GRID, or gay-related immune deficiency. The term AIDS was later introduced in 1982. In 1983, two separate research groups, led by Robert Gallo and Luc Montagnier, both came to the conclusion that the HIV virus was the infectious agent responsible for AIDS. HIV could be an evolved form of the SIV virus that infects wild chimpanzees. The earliest well-documented case of HIV in a human is from 1959 in the Congo, and HIV may have been in the U.S. as far back as 1966. Most HIV infections can actually be traced back to a single individual who was infected in Haiti, then brought the infection to the U.S. around 1969. Those infected with HIV also have to face rejection and discrimination because of their condition. About 600,000 people in the U.S. have died from AIDS-related causes. 1.1 million Americans are living with the HIV virus, but an estimated 18% are living undiagnosed. AIDS both impacts and is impacted by society and lifestyle. It is passed on sexually or by sharing needles, showing that it can come from a lifestyle of drugs and promiscuity, a common way of life for some young adults. Those infected cannot have children without passing on the disease, deterring them from living the life that they wanted. When AIDS was first introduced, no one believed that they could get it, but once well-known public figures like Magic Johnson and Freddie Mercury had contracted it, people became aware that they too are at risk.
The HIV virus that I have attained, I will have to retire from the Lakers.
Even after more than 30 years since its recognition, AIDS is still a huge concern and has affected countless lives of Americans everywhere.
The goal of the Human Genome Project was to sequence all 3 billion bases of the human genome. The United States Department of Energy, curious of the effects of radiation on the genome, established an early genome project in 1987. While the idea had already been introduced, planning for the project would not be complete until 1990. It was initially believed that it would take 15 years to complete the sequencing, but new technology like restriction fragment length polymorphisms, the polymerase chain reaction, and pulsed field gel electrophoresis allowed for faster progression. By 2001, 90% of the genome had been sequenced, but it was not until April 2003 that the largest collaborative biological project was officially complete. The Human Genome Project revealed that there are only about 20,000 human genes, contrary to the previous belief that there could have been as many as 140,000 genes. The research on the genome would later aid the study of how to treat, prevent, and cure disease. Having access to DNA sequence would also help in understanding viruses, identifying mutations, and researching cancer. Decoding the genome also came with major social implications. The vast information gained from learning the complete sequence of someone's genome could create new prejudices based on a person's genetic code. The mapping of the genome was not the only accomplishment in this venture, but also the development of technological improvements that accompanied the project. Without the project, there would be no drive to invent new biological tools. Sequencing the genome helped the scientific community make even more progress and go beyond what was previously imagined.